Therapeutic benefit of combining calorie-restricted ketogenic diet and glutamine targeting in late-stage experimental glioblastoma. Abstract. Glioblastoma, GBM, is an aggressive primary human brain tumor that has resisted effective therapy for decades. Although glucose and glutamine are the major fuels that drive GBM growth and invasion, few studies have targeted these fuels for therapeutic management. The glutamine antagonist, 6-diazo-5-oxo-L-norlusine, DON, was administered together with a calorically restricted ketogenic diet, KDR, to treat late-stage orthotopic growth in two syngeneic GBM mouse models, VMM3 and CT2A. DON targets glutaminolysis, while the KDR reduces glucose and, simultaneously, elevates neuroprotective and non-fermentable keton bodies. The diet-drug therapeutic strategy killed tumor cells while reversing disease symptoms and improving overall mouse survival. The therapeutic strategy also reduces edema, hemorrhage, and inflammation. Moreover, the KDR diet facilitated DON delivery to the brain and allowed a lower dosage to achieve therapeutic effect. The findings support the importance of glucose and glutamine in driving GBM growth and provide a therapeutic strategy for non-toxic metabolic management. Introduction. Glioblastoma, GBM, has the highest mortality rate among primary brain tumors in adults and remains largely unmanageable with a life expectancy of about 6 to 24 months following diagnosis. Despite decades of research, little progress has been made in improving GBM survival. 1. A defining characteristic of GBM is the secondary structures of Schura, which includes diffuse parenchymal growth and invasion over the subpile surface, along white matter tracks, and through the Virchow-Robin space. 2. The highly invasive nature of GBM makes most current therapies ineffective. Most patients die from intracranial pressure due to inflammation, edema, and distal tumor cell invasion. Hence, the proliferative and invasive nature of GBM is the major hindrance for effective therapeutic intervention. GBM contains a range of morphologically diverse neoplastic cell types that express glial, stem cell, and myeloid mesenchymal markers 3, 4, 5. Also recognized are abnormalities in the number, structure, and function of mitochondria in GBM tumor tissue 6, 7, 8, 9, 10. In addition, recent studies show abnormalities in GBM mitochondrial-associated membranes, which are also critical for normal mitochondrial function. 11. Moreover, the content and composition of cardiolipin, the signature lipid of the mitochondrial inner membrane that regulates oxidative phosphorylation, OXPHAS, shows deviations from the normal in 5 different murine GBM models. 12. As mitochondrial function and efficiency is dependent on structure. 13. OXPHAS is expected to be lower in GBM than in normal brain tissue. Indeed, significant evidence shows that OXPHAS is defective in human and murine GBM 7, 12, 14. Regardless of GBM cell type, glucose and glutamine are required for growth and invasion through glycolysis and glutaminolysis, respectively 15, 16, 17, 18, 19. Although glucose is the primary metabolic fuel for neurons and glia, normal brain cells can transition to keton bodies, primarily hydroxybutyrate, for energy under hypoglycemic conditions. 20. In contrast, GBM cells lack metabolic flexibility and are susceptible to death in response to reduced glucose bioavailability. 21. Although brain glutamine levels are tightly regulated via the glutamine-glutamate cycle, the bioavailability of glutamine increases following surgery, radiation, and chemotherapy, which fuels tumor growth. 2022. If glucose and/or glutamine remain available, the tumor cells will utilize the fuel and grow, making long-term management difficult. The calorically restricted ketogenic diet, KDR, is a low-carbohydrate, high-fat diet that reduces blood glucose and increases blood keton bodies to therapeutic levels, while simultaneously inducing anti-inflammatory effects. 23. The primary keton bodies, hydroxybutyrate and acetoacetate, are also neuroprotective and non-fermentable. 24-25. We found previously that the KDR is a non-toxic anti-inflammatory approach to reduce pathology in experimental human and mouse brain tumors. 23. The neoplastic GBM cells can also synthesize high-energy phosphates through mitochondrial substrate-level phosphorylation supported by glutaminolysis. 25-26. In addition, glutamine provides a continuous source of nitrogen for de novo synthesis of nucleotides and proteins in cancer cells. 27. The extent of glucose and glutamine metabolism in tumor cells depends not only on the tumor cell type, but also on the location and microenvironment. Recently, the efficacy of simultaneous inhibition of both glycolysis and glutaminolysis has been demonstrated in experimental cancer models and in a human GBM case report. 5. Therapeutic success depends on modifications in dosage, scheduling, and timing in order to enhance efficacy while reducing toxicity. 6. 6-diazo-5-oxo-L-norlusine, DON, is a glutamine antagonist that was initially isolated from a Streptomyces broth in 1956. DON blocks multiple glutaminases, thus restricting key metabolites needed for glutaminolysis and the synthesis of nucleotides and proteins. The antitumor efficacy of DON was confirmed in different tumor models, and in humans with various cancers. In the present study, we show a unique therapeutic response from the simultaneous targeting of glucose and glutamine using a KDR and DON, respectively, in syngeneic orthotopic mouse models of GBM. The VMM3 GBM tumor arose in the cerebral cortex of a mouse from the VMDK inbred strain, which has a high incidence of spontaneous brain tumors relative to other strains. The mesenchymal VMM3 tumor cells invade distally throughout the brain using the secondary structures of SHRA2. In addition to the VMM3 tumor, we also evaluated the effects of the KDR and DON combination on the growth of the CT2A brain tumor. The highly angiogenic CT2A tumor was produced from 20-methancholanthrene in the cerebral cortex of a C57BL6J mouse, and has several characteristics in common with the neoplastic neural stem cells found in human GBM. Abnormalities in the electron transport chain were found in both tumors. Our data show that the combination of KDR and DON administration cause massive tumor cell death in both of these preclinical GBM mouse models and highlights the importance of targeting glycolysis and glutaminolysis simultaneously for the metabolic management of GBM. Results. The aim of this research was to determine if the simultaneous targeting of glucose and glutamine, while under therapeutic ketosis, could manage late-stage growth and enhance survival without toxicity in two different syngeneic mouse models of GBM. We administered DON 7 days after tumor implantation, which is considered late for the VMM3 FLUC tumor, as neurological signs and symptoms first appear at 10 to 12 days post-implantation, with morbidity occurring in 100% of the tumor-bearing mice by 14 to 18 days too. Additional results were obtained in C57BL6J inbred mice bearing the syngeneic CT2A high-grade stem cell glioma 32. DON, at the concentrations used in this study, had no toxic effects in non-tumor-bearing VMDK mice, as was shown previously for C57BL6J mice. To observe the effect of the KDR and DON on the VMM3 tumor model, we conducted 3 consecutive experiments. Experiment 1 included 4 groups: SDUR plus DON, KDR plus DON, with lower DON dosages, 0.1 and 0.5 mg per kilogram. Experiment 2 involved both in vivo and ex vivo imaging of 2 study groups, KDR and KDR plus DON, following the administration of a higher dose of DON, 1 mg per kilogram. Experiment 3, a survival study, also used the higher DON dose, 1 mg per kilogram, and included the 4 groups from Experiment 1, SDUR plus DON, KDR plus DON, Figure 1. Figure 1. Restricted ketogenic diet with DON reduces progression and mortality of the VMM3 GBM. VMDK inbred mice were implanted orthotopically with a small, 1 mm x 1 mm, tissue fragment from the VMM3 tumor on day 0. The implanted mice were divided into 2 groups on day 4 and were fed either a standard chow diet unrestricted or ad libitum, SDUR, or a ketogenic diet, KDR, in restricted amounts to reduce body weight by about 15%. DON, 0.1 to 1.0 mg/kg, was injected IP 7 days following orthotopic tumor implantation. The diet feeding was continued and DON was injected every day or every alternate day as shown in A. All mice were imaged in vivo and terminated on day 14 or 15 when all control mice appeared moribund. Experiments 1, 2. For Experiment 1, bioluminescence was scored from 0 to 4, following the administration of lower doses of DON, 0.1 and 0.5 mg per kilogram. Values are expressed as the mean plus or minus SEM, and a one-way analysis of variance followed by Tukey's post-hoc test was performed to determine the significance between groups. B. In Experiment 2, in vivo bioluminescent photon values were obtained following the administration of DON, 1 mg per kilogram, in mice under the KDR, C. The average in vivo bioluminescent photon values were calculated for the KDR, n equals 4 mice, and KDR plus DON, n equals 8 mice, in Experiment 2. Values are presented as the mean plus or minus SEM, and the p-value was calculated using a two-tailed student's t-test, D. In vivo bioluminescent images of 3 representative mice from both study groups in Experiment 2 are shown in E. For Experiment 3, a survival study was performed and a Kaplan-Meier survival plot was configured for SDUR, n equals 15 mice, SDUR plus DON, n equals 10 mice, KDR, n equals 10 mice, and KDR plus DON, n equals 10 mice, F. The log-rank statistical analysis test showed a significant difference between groups in this survival study. Source data are provided as supplementary data 1. Full-size image. KDR plus DON reduces VMM3 burden and increases survival. The KDR plus DON treatment was performed as outlined in the study timeline, Figure 1a. In Experiment 1, 2 doses of DON, 0.1 and 0.5 mg per kilogram, were used in both the SDUR and KDR groups, Figure 1b. Deluciferin was injected intraperitoneally, IP, 15 days after tumor implantation, and in vivo bioluminescence imaging was performed. The total VMM3 tumor bioluminescence photon values were scored from 0 to 4, with 0 being the baseline value and 4 being the highest value. Several mice from the SDUR group that did not receive DON had a maximum photon score of 4, whereas the mice from the KDR group plus DON had a minimum photon score of 0 or 1. Compared to the control SDUR group, mice receiving the KDR alone or SDUR plus DON had a 19 and 44% lower photon value, respectively, Figure 1b. The bioluminescence photon score was 75% lower in mice that received the KDR plus DON than in mice receiving each therapy alone, indicating a synergistic effect of the diet-drug combination. Experiment 2 involved a larger number of mice, n equals 8 mice, that were treated with a higher dose of DON, 1 mg per kilogram. Due to the observed synergy between the KDR and DON in experiment 1, only the KDR group was selected for further experiments. All mice in this experiment expressed tumor burden by day 7. Individual brain bioluminescent values are shown in Figure 1c. A baseline value of bioluminescence was seen in 7 out of the 8 KDR-fed mice that received DON, and a reduction in average photon value was observed in the DON-treated mice, Figure 1d. A representative number of mice from both study groups are shown in Figure 1e. All untreated SDUR control mice died between day 15 and 18. In contrast, mice treated with the diet-drug combination survived until day 40 and onwards, Figure 1f. A standard diet-restricted SDR group or a ketogenic diet unrestricted KDUR group were not included in this study. We previously showed that a KDR is more effective at reaching the target level of blood glucose and ketones than is a SDR 23, 34, 35. In other words, the keton levels are higher and the glucose levels are lower under KDR feeding than under SDR feeding despite similar caloric restriction and reduced body weight. A KDUR was also not included in this study, as consumption of ketogenic diets in unrestricted amounts can cause insulin insensitivity and weight gain, as we previously described 23, 35, 36. KDR plus DON reduces ex vivo bioluminescence in VMM3 brain. The VM mice from experiment 2 were sacrificed after in vivo imaging, and their brains were immediately resected and individually imaged ex vivo for total bioluminescence as described in the methods. Ex vivo imaging provides a more accurate assessment of tumor progression than in vivo brain imaging due to the direct contact of the D-luciferin with the brain tissue and the removal of the blood-air-brain barrier as a limiting factor. The individual ex vivo bioluminescent values for the DON-treated mice were below the baseline value, indicating a reduction in tumor cell viability, Figure 2a. These values were consistent with the in vivo photon values, Figure 1c. A reduction in average ex vivo photon value was also observed in the mice treated with DON alone, Figure 2b. A representative number of brains from the KDR and the DON-treated mice are shown in comparison to the SDUR mice, Figure 2c. It should be noted that an accurate in vivo bioluminescent value was not obtained for mouse number 3 and number 6 due to several variables during the imaging process: tumor burden, isofluran exposure, blood collection, etc. If an individual mouse did not provide an accurate in vivo bioluminescent reading, the brain was immediately resected, placed in sterile PBS and imaged ex vivo. For this reason, there are slight discrepancies in the number of mice imaged for in vivo bioluminescence and for ex vivo bioluminescence in experiment 2. Figure 2. Restricted ketogenic diet with DON reduces VMM3 GBM bioluminescence. VMDK mice were implanted with a VMM3 tumor, as described in Figure 1. Mouse brains were imaged ex vivo on day 14 or 15. Mice were imaged ex vivo after in vivo bioluminescent imaging, as described in Figure 1. Individual ex vivo bioluminescent photon values were measured following the administration of DON, 1 mg per kilogram, in KDR mice, A. From the individual measurements, the average ex vivo bioluminescent photon value was calculated for both the KDR, n equals 6 mice, and KDR plus DON, n equals 8 mice, groups. Values are presented as the mean plus or minus SEM, and significance of differences was determined following a two-tailed student's t-test, B. Ex vivo bioluminescent images of brains from representative mice in experiment 2 are shown in comparison to SDUR, C. Source data are provided as supplementary data 1. Full-size image. KDR plus DON reduces proliferation and kills VMM3 cells. Histological evaluation of brain tumor tissue from mice with the VMM3 tumor is shown in Figure 3. Compared with the diffuse, ill-defined border of the VMM3 tumor cells growing in the SDUR brains, the tumor in the KDR brains appeared less dense, less invasive, and had a more defined border, Figure 3a. DON-treated brain tumors showed an area of dead cells in the primary tumor. In addition, the percentage of Ki67 proliferating cells, green, was lower in the KDR tumor than in the SDUR tumor and was further reduced in the KDR plus DON-treated tumor tissue, Figure 3b. Figure 3. Restricted ketogenic diet with DON kills VMM3 GBM cells in brain. VMDK mice were implanted with VMM3 cells orthotopically in the brain on day 0 as described in Figure 1. The brains were fixed in formalin for histology, processed, and stained as described in methods. Histological analysis, H&E, was used to validate the presence of tumor cells, A. The top panel images show the core of the tumor mass growing in the brain. The black boxes in the top panel's images are shown in higher magnification in the bottom panels. All scale bars are 150 micrometers for x 175 micrometers for x 200. Ki67 positive nuclear staining, times 100, as expressed by green fluorescent labeled cells are shown in B. DARPi is used for the Ki67 negative nuclear stain. The invasion of tumor cells in the brain tissue is evident, arrows, in both histological, H&E, analysis, times 100, and Ki67 staining, C. All scale bars for B and C are 100 micrometers. Full-size image. VMM3 cells are highly invasive through the secondary structures of Schura2. VMM3 cells were observed invading from the core tumor into the normal-appearing brain, supplementary Figure 1. Moreover, the extensive perivascular necrosis and subarachnoid invasion, hallmarks of human GBM, supplementary Figure 1, were clearly present in the VMM3 model of GBM. The KDR reduced the invasion of VMM3 cells as shown in both H&E and Ki67 staining, figures 3b, c. VMM3 GBM tumor cells were seen invading the perivascular area in the SDUR brain, whereas fewer invading GBM tumor cells were seen in the KDR brains. No invading VMM3 cells were seen in the KDR plus DON-treated brains, figure 3c. These findings indicate that the diet drug therapy both kills tumor cells and inhibits invasion. KDR facilitates DON delivery to the VMM3 tumor. Plasma and brain tissues were prepared for DON analysis 1 hour after systemic DON injection, as described in methods. LC-MS analysis showed that the concentration of DON in the VMM3 tumor tissue was about twofold greater in mice fed the KDR than in mice fed the SDUR, figure 4a. In another independent experiment, using a similar analytical system, the concentration of DON in the VMM3 tumor tissue was about threefold greater in mice fed the KDR than in mice fed the SDUR, figure 4b. In both methods of measurement, DON delivery to the brain was greater in the mice fed the KDR than in the mice fed the SDUR, figure 4a, b. DON was detected in brain tissue 60 minutes following IP injection. These findings indicate that the KDR facilitated delivery of DON to the VMM3 tumor tissue. Figure 4. Restricted ketogenic diet increases DON delivery to the VMM3 GBM. The content of DON in the VMM3 brain tumor tissue was quantified using two LC-MS procedures, as described in methods. The brain tissue was analyzed for DON content 60 minutes after IP DON injection. The values in experiment A are presented as mean of two independent samples, while the values in experiment B are presented as the mean plus or minus SEM, n equals 3 mice. Following a two-tailed student's t-test, the difference between the two groups in experiment B was significant, p less than 0.05. Both analytical procedures showed that DON content in tumor tissue was greater under KDR feeding than under SDUR feeding. Source data are provided as supplementary data 1. Full-size image. KDR plus DON reduces TNF expression and GKI. TNF expression was measured in lysates prepared from the right brain cortex that contained the VMM3 tumor. These mouse brain samples are different from the mice used for imaging in figures 1 and 2. A high level of TNF expression, a biomarker for inflammation, was found in the right brain cortex of mice fed the SDUR. Two independent analyses showed a similar trend in the TNF level, pg milligram of protein in the lysate. TNF expression was either low or undetectable in the normal brain tissue, figure 5a. TNF expression was lower in the brain of KDR-fed mice than in the brain of SDUR-fed mice, whereas TNF expression was even lower in the KDR plus DON-treated mice, suggesting a minimum amount of tumor load and reduced inflammation compared to the other two groups, figure 5a. Figure 5. Restricted ketogenic diet with DON reduces TNF in the VMM3 brain tumor tissue, and the glucose-keton index, GKI, in the blood. ELISA was used to measure TNF in brain tumor tissue lysates in two different experiments, and the content was expressed as pg milligram of protein. Normal brain, NB, was used as a negative control tissue. The range of TNF concentration was minus 0.04 to 0.1 pg milligram for NB, n equals 2 normal mouse brain tissue, 0.4 to 14.7 pg milligram for SDUR, n equals 5 mouse tumor brain tissue, 0.09 to 11.2 pg milligram for KDR, n equals 5 mouse tumor brain tissue, and 0.02 to 1.0 pg milligram for KDR plus DON, n equals 7 mouse tumor brain tissue, A. Data showing that blood glucose is lower and blood ketones are higher in mice fed the KDR or KDR plus DON, n equals 5 independent mouse blood samples per group, than in mice fed the SDUR, n equals 10 independent mouse blood samples. This shift in blood glucose and ketones causes a significant reduction in the GKI, B. Values are expressed as the mean plus or minus SEM, and a one-way analysis of variance followed by Tukey's post-hoc test was performed to determine the significance between groups. Source data are provided as supplementary data 1. Reduced glucose and increased keton body levels are blood biomarkers for the KDR and evidence of therapeutic ketosis. The ratio of glucose to keton bodies, glucose-keton index, GKI, has been linked to the therapeutic efficacy of the ketogenic diet, KD, for brain cancer 36. The lower the GKI value, the greater the metabolic stress on the tumor cells. The GKI values for the KDR mice, treated and untreated with DON, were lower than for SDUR mice. The GKI values in the KDR plus DON mice were similar to that in the KDR mice, indicating that a low GKI was maintained under DON treatment, figure 5b. KDR plus DON reduces IBA1 expression in VMM3. IBA1 protein expression was analyzed in the VMM3 brain tumor tissue by immunohistochemistry and Western blot. IBA1 was heavily expressed in VMM3 tumor and normal microlia macrophages, as we described previously 37. The KDR reduced the expression of IBA1 as shown in both immunohistochemistry and Western blot, figure 6a, b. There was a reduction in IBA1 expression in DON-treated brain tumor tissue. It is important to note that the majority of tumor cells in the DON-treated tumors were dead and therefore did not stain positively for nuclear methyl green, figure 6a. This finding is consistent with the histological observation found from our H&E analysis, figure 3. The reduced IBA1 staining is also consistent with the reduced TNF expression, figure 5, suggesting reduced inflammation in the brains of both KDR and DON-treated mice. The reduced IBA1 histological staining was also consistent with the findings from Western blot analysis of IBA1 expression, figure 6b, c. Expression of IBA1 in the brain tissue of the KDR plus DON-treated mice was lower than the expression in the untreated SDUR mice, and was similar to the expression in normal non-tumor mouse brain. IBA1 expression was also lower in the KDR mice than in the SDUR mice, but the reduction was not significant, fig. 6b, c, and supplementary figure 2. Figure 6. Restricted ketogenic diet with DON reduces the IBA1 expression in the VMM3 brain tumor tissue. Immunohistochemistry, IHC, of IBA1 was performed in formalin-fixed brain tissues as described in methods. In comparison to all other groups, the expression of IBA1 is highest in the SDUR tumor as seen by the positively stained brown cells. IBA1 IHC staining was noticeably less in the VMM3 tumor than in the KDR-fed mice and the DON-treated mice, A. All scale bars are 100 micrometers. Western blot analysis of IBA1 protein expression in the tumors indicates a decrease in the expression of IBA1 in the DON-treated tumors, N equals 3 mouse tumor brain tissue, in comparison to SDUR, N equals 3 mouse tumor brain tissue. A decrease in expression was also seen for KDR mice, N equals 2 mouse tumor brain tissue. Normal brain, NB, was used as a negative control tissue, B and C. Source data are provided as supplementary data 1. Full-size image. KDR plus DON reduces edema in CT2A and increases survival. The CT2A tumor is characterized as a highly angiogenic malignant mouse astrocytoma with stem cell characteristics 32-38. The wet weight of the brain plus CT2A tumor was lower in mice fed the KDR than in mice fed the SDUR, indicating a reduction of edema, figure 7a. Ex vivo bioluminescence of brains containing the CT2A tumor was lower in mice fed the KDR plus DON than in mice fed the SDUR, indicating a reduction in the number of living CT2A tumor cells in the brain, figure 7b. Representative mice with the CT2A brain tumor are shown in figure 7c. The brains of mice bearing the CT2A tumor that were fed the SDUR were highly hemorrhagic, thrombotic, and edematous. In contrast, the brains of CT2A bearing mice that were treated with KDR plus DON appeared normal without hemorrhage, thrombosis, or edema, figure 7c. All untreated SDUR control mice containing the CT2A tumor died between day 10 and 13, figure 7d. In contrast, CT2A bearing mice treated with the diet drug combination survived until day 24 and onwards. Histological analysis also showed that CT2A tumor cell density, mitotic figures, and hemorrhage was less prevalent in the mice fed the KDR than in the mice fed the SDUR, supplementary fig. 3. The appearance of mitotic arrest or catastrophe was also seen in the CT2A tumor of mice treated with the KDR plus DON, supplementary figure 3. Figure 7. Restricted ketogenic diet with DON reduces progression and mortality of the CT2A GBM. C57BL6J mice were implanted with CT2A tumor fragments on day 0, as described in figure 1. Brain wet weights were measured for SDUR, N equals 4 mouse brains, KDR, N equals 7 mouse brains, and KDR plus DON, N equals 5 mouse brains, A. Ex vivo bioluminescent photon values were calculated for the same brain samples and presented as the mean plus or minus SEM, B. All brains were imaged on day 14 or 15. A one-way analysis of variance followed by Tukey's post-hoc test was performed to determine the significance between groups. Representative brain sample images from each study group were portrayed in C. A Kaplan-Meier survival curve was plotted for the SDUR, N equals 6 mice, SDUR plus DON, N equals 8 mice, KDR, N equals 6 mice, and KDR plus DON, N equals 8 mice, groups, D. The log-rank test was used to determine the significance. Source data are provided as supplementary data 1. Full-size image. Discussion. This study documents the therapeutic action of glucose and glutamine targeting in the VMM3 and CT2A syngeneic mouse models of GBM. It is known that calorie restriction, ketogenic diets, and DON can reduce metabolites in the glycolytic and glutaminolysis pathways, respectively 33, 39, 40, 41. However, We showed that the KDR plus DON could rescue mice with these GBM tumors from late-stage orthotopic growth. The diet drug therapy reduced brain swelling, hemorrhage, and morbidity. The KDR alone reduced the VMM3 flux cell invasion and proliferation as seen by both in vivo and ex vivo bioluminescent imaging of the brains, with Ki67 staining, and with histology of the brain tissues. Moreover, the KDR produced therapeutic ketoses, low GKI value, and reduced TNF and IBA1 levels, suggesting reduced inflammation as compared to the SDUR control brains. The ability of DON treatment to arrest tumor growth and promote survival late in disease progression suggests a potentially direct role for glutamine metabolism in VMM3 flux growth and invasion. This finding is consistent with the importance of glutamine in the metabolism of mesenchymal cells, from which the VMM3 tumor is derived. Although DON treatment alone was effective in reducing tumor growth and invasion in the control SDUR mice, a synergistic effect was observed when DON was administered together with the KDR, providing evidence for a diet-drug interaction. The findings also support the press-pulse therapeutic strategy for the metabolic management of cancer, with the KDR serving as a press therapy and DON serving as a pulse therapy 6. Interestingly, the level of DON in the brain tumor tissue was higher under KDR feeding than under SDUR feeding, suggesting that the KDR facilitates DON delivery to the brain. This is significant because it allowed for a lower therapeutic dose of DON when administered under a KDR regimen. A similar observation was made previously in showing that a KDR facilitated the delivery of the immunosygr, N-butyl-deoxynogerimisin, across the blood-air-brain barrier in mice with Sandhoff disease 43. Although the mechanism remains to be determined, these findings suggest that the KD has potential as a facilitator of drug delivery to the brain for a broad range of neuropathological conditions. It is also important to mention that our previous findings clearly demonstrated that caloric restriction can maintain the blood vessel integrity in the mouse CT2A and the human U87 GBM and thus reduce the leakiness of the neovasculature 44. Interestingly, a recent observation by Gordon et al. Showed that DON treatment promotes the recovery of blood-air-brain barrier integrity in the mouse model of cerebral malaria. 33. Further studies are needed to clarify the mechanism by which a KDR facilitates drug delivery to the brain. DON has shown therapeutic efficacy in human cancers of the blood, colon, and lung, but issues of toxicity were noted in some cases. 30. No noticeable toxicity was seen in the DON-treated mice until the end of the study, when mild muscle loss was observed in some of the mice. It is proposed that issues with DON toxicity arise when there is an appreciable competition for glutamine between host and tumor. There is also increasing evidence supporting the protective role of glutamine supplementation for cancer patients. 45. Since tumor consumption of glutamine is dissipative, glutamine supplementation might not increase tumor growth. 46. However, additional preclinical studies will be needed to determine if glutamine supplementation could be used together with DON. It is our view that strategies for managing toxicity of a therapeutically effective drug would be as important as developing a new drug. Our results indicate that the KDR enhances the therapeutic action of DON, thus reducing dosage and toxicity. The therapeutic action of the diet drug effect seen in the VMM3 tumor was also seen in the CT2A neural stem cell tumor. As observed from histological analysis, the KDR plus DON treatment caused massive mitotic arrest or catastrophe in the CT2A brain tumor cells. The results show that therapeutic efficacy against orthotopic brain tumor growth was greater using the diet drug combination than in using either the KDR or DON alone. The CT2A tumor shares several characteristics with glioma stem cells 32, whereas the VMM3 tumor shares several characteristics with the invasive mesenchymal cell found in most GBM2. Hence, our findings suggest that the KDR plus DON therapeutic strategy could potentially target the two most common neoplastic cell types found in human GBM3. Our recent human GBM case report provides support for our prediction. 15. Several mechanisms could underlie the therapeutic action of the diet drug therapy used to treat the VMM3 and CT2A tumors. The KDR will simultaneously target the glycolytic and pentose phosphate pathways, which are upregulated in GBM 47, 48, 49. We previously showed that calorie restriction and restricted ketogenic diets target the IGF-1, PI3K, AKT, and HIF-1 alpha signaling pathways in the CT2A tumor 3850. The downregulation of these pathways would reduce angiogenesis, inflammation, and mTOR signaling, while enhancing tumor cell apoptosis 23, 44, 51, 52. As glucose is the fuel for glycolysis, the pentose phosphate pathway, and serine biosynthesis, the KDR should reduce multiple growth metabolites, tumor cell glutathione levels, and nucleotide synthesis. Furthermore, the KDR should also reduce 1-carbon metabolism, especially in glioma cells lacking OXPHAS capacity 25, 49, 53. Hence, the glucose-restricting action of the KDR will target multiple signaling pathways linked to glioma growth and progression. In addition to restricting glucose availability, the KDR will also elevate hydroxybutyrate and acetoacetate, the major circulating ketone bodies produced in the liver, from the active metabolism of medium-chain triglycerides 54. Ketone bodies increase the redox span of the CoQ couple, thus reducing oxidative stress in normal brain cells 54. These keton bodies are also neuroprotective and have beneficial therapeutic value for various diseases. 2455. As keton bodies are non-fermentable and require efficient OXPHARs for generating ATP 2554, the VMM3 and CT2A cells will be unable to metabolize these metabolites for energy due to insufficient OXPHARs. 1225. Keton bodies cannot be used as an alternative energy fuel in cells with defective mitochondria. 1225. Previous studies demonstrated that glioma cells cannot use keton bodies and that keton bodies do not stimulate tumor growth. 5657. Hence, the KDR has a dual function in 1. Targeting glucose-dependent signaling pathways that drive tumor growth, and 2. In providing an alternative metabolic fuel to normal brain cells under glucose restriction. While the KDR will reduce metabolites through glycolysis and the pentose phosphate pathway, DON will block glutaminolysis, thus depriving the tumor cells of the amide nitrogen needed for nucleotide and protein synthesis, and at the same time, will deplete the glutamate needed for synthesis of ketoglutarate, kilogram, 255859. Kilogram is a precursor for lipid synthesis through reductive carboxylation and is the substrate for ATP synthesis through the succinate-CoA ligase reaction in the TCA cycle under hypoxia. 2560. As mentioned previously, however, glutamine targeting is more challenging than is glucose targeting due to the importance of glutamine for the immune system and the gut. 661. Consequently, glutamine targeting must be done strategically to avoid damage to those systems that are needed for normal physiological function. 615. It is for this reason that we administered DON to the tumor-bearing mice at low doses and transiently. We administered DON 7 days after tumor implantation, which is considered late for the VMM3 FLUC tumor because neurological signs and symptoms start to appear 10 to 12 days post-implantation followed by 100% death in 14 to 18 days. The late administration of DON was chosen to ensure adequately high keton levels were achieved in order to facilitate neuroprotection and to reduce tumor inflammation. Secondly, the late treatment could serve as a comparable model when treating late-stage GBM patients. A 60 to 70% reduction in tumor growth was observed when DON was administered at 0.5 mg per kilogram once or twice per week. In comparison, a near-complete resolution of the brain tumor was achieved when 1 mg per kilogram of DON was administered with the same schedule, as seen with bioluminescent imaging. These results are significant because no other clinical or preclinical study using DON considered a KDR as a method for protecting the host from toxicity. Case reports of GBM and other metastatic cancers show the significance of the KDR when combined with other therapies. Our findings support previous suggestions that reduced glutaminolysis would be a key therapeutic action of DON against the growth of glutamine-dependent tumors, including GBM. Although it is known that DON targets glutaminolysis in tissues and that reduced levels of circulating glucose will reduce glycolysis, we were unable to find consistent brain metabolite changes in pathways of glycolysis and glutaminolysis in a preliminary metabolomic analysis of KDR plus DON-treated VMM3 mouse brains. Due to the confounding variables in the in vivo environment, dead tumor cells, necrotic tissue, and various types of host-infiltrating cells, we believe that an in vitro analysis in defined media could provide a clearer picture of the changes in metabolites of the glucose and glutamine pathways due to DON or other drug treatments in the VMM3 and CT2A cells. It will also be important to determine if other glutaminolysis inhibitors, e.g., epigallocatechin 3 gallate, EGCG, BIS2-5-phenylacetamide 1,2,4-thiodiazole 2-YL, ethyl sulfide, BPTEs, and CB839. Can match the therapeutic action of DON against brain cancer when used together with the KDR pretreatment with a ketogenic diet before implementing the standard of care is suggested in order to protect normal brain cells from treatment toxicities. 22. It is important to mention that our findings in the VMM3 and CT2A syngeneic GBM models might be considered contradictory to some previous studies in GBM patients and in experimental systems. Previous studies from Bachu and colleagues suggest minimal 13C glutamine anaplerosis in GBM, implying that glutamine restriction in this context may not be therapeutic. 68. As DON inhibits multiple glutaminases, it is possible that DON does not directly impact glutamine anaplerosis in vivo, but acts on other related pathways and systems. We recently described how glutamine is the only amino acid not requiring an energy investment to generate ATP through mitochondria substrate-level phosphorylation, MSLP. Tardito et al. Found that under glutamine starvation, intracellular oleate was unaffected, and glucose-dependent glutamate production increased, implying that the contribution of glutamine to growth is largely independent of anaplerosis. 6. Further, Oisel et al. Identified 2 clusters of GBM that are glutamine high and glutaminolo based on glutamine utilization. These investigators also mentioned that the most aggressive GBM contained mesenchymal cells that utilized the highest levels of glutamine. The VMM3 cells are highly invasive and of mesenchymal origin. Nevertheless, we use caution in the interpretation of our data, as the metabolic effects of DON's action in killing the GBM cells were inferred rather than measured directly. In the present study, we demonstrate the benefits of targeting glutamine under a KDR for managing experimental GBM. The influence of targeting both glucose and glutamine on the metabolic pathways involved with GBM growth is shown in Figure 8. This treatment was capable of arresting the growth of tumor cells and in promoting the survival of mice with 2 different syngeneic GBM tumors grown orthotopically. As the mice used for these studies were not treated with surgery, radiation, or standard chemotherapy, it is unclear if a similar therapeutic response would be seen in GBM patients using this approach together with current standard of care. Because of the high mortality following a GBM diagnosis, our findings should have clinical implications, especially in light of our recent case report. Furthermore, our studies reveal a potential role for the ketogenic diet in facilitating drug delivery to brain tumors. Figure 8. Targeting glucose and glutamine using KDR with DON for the metabolic management of the VMM3 and CT2A experimental GBM. GBM tumor cells are largely dependent on glucose and glutamine for survival and growth. Energy through substrate-level phosphorylation, SLP, in the cytoplasm, glycolysis, and in the TCA cycle, glutaminolysis, will compensate for reduced energy through oxidative phosphorylation, OXPHAS, or hypoxia that occurs in these GBM cells. The KDR will reduce glucose carbons for both the glycolytic and pentose phosphate, PPP, pathways that supply ATP and precursors for lipid and nucleotide synthesis, as well as for glutathione production. DON will inhibit glutaminases, thus depleting glutamate and the glutamine-derived amide nitrogen for ammonia and nucleotide synthesis. This diagram and legend have been modified from that presented previously under the terms of the Creative Commons Attribution 4.0 International License in Nutrition and Metabolism by Thomas Seyfried et al. This diagram and legend have been modified from that presented previously under the terms of the Creative Commons Attribution 4.0 International License in Nutrition and Metabolism by Thomas Seyfried et al. Materials and methods. Mice. Mice of the VMDK, VM, strain were obtained as a gift from H. Fraser, University of Edinburgh, Scotland. The C57BL6J, B6, mice were obtained originally from the Jackson Laboratory, Bar Harbor, Maine. All mice used in this study were housed and bred in the Boston College Animal Care Facility using IACUC-approved husbandry conditions 28. Male and female mice between 8 to 10 weeks of age were used for all studies. All animal procedures and protocols were in strict accordance with the NIH Guide for the Care and Use of Laboratory Animals and were approved by the Institutional Animal Care Committee at Boston College under Assurance Number A390501. The VMM3 and CT2A murine GBM models. The VMM3 tumor used in this study arose spontaneously in the cerebrum of an adult male mouse of the VMDK inbred strain. A cell line was prepared from the VMM3 tumor 37. The VMM3 tumor manifests all of the invasive characteristics seen in human GBM2. The CT2A tumor was originally produced from implantation of 20-methylcholanthrin into the cerebral cortex of a C57BL6J mouse and was broadly classified as a poorly differentiated highly malignant anaplastic astrocytoma, and a cell line was produced from this. Tumor 31. Both cell lines are mycoplasma-free and we use Mycozap, Lonza, kit to eradicate mycoplasma. More recent studies have classified the CT2A tumor as a neural stem cell tumor 32. The VMM3 and the CT2A cell lines were transduced with a lentivirus vector, gift from Miguel Sena Estevez, containing the firefly luciferase gene under control of the cytomegalovirus promoter, VMM3 FLUC, to produce VMM3 FLUC and CT2A FLUC cell lines 37. This transduction allows the cells to be tracked in the brain using bioluminescent imaging. Tumor implantation. Tumor implantation was performed using our standard protocol 2. Briefly, mice were anesthetized with isofluran, 5% in oxygen. The tops of the heads were disinfected with ethanol and a small incision was made in the scalp over the midline. A 3 mm 3-burr hole was made in the skull over the right parietal region behind the coronal suture and lateral to the sagittal suture. Small, 1 mm 3, tumor fragments were implanted approximately 1.5 to 2.0 mm deep into the cortical region using a trocar, as previously described. The skin flaps were closed with 7 mm reflex clips. The mice were placed in a warm room, 24 degrees Celsius, until they were fully recovered. The procedure confirms 100% recovery within a few hours of implantation. The GBM tumor cells are highly invasive regardless of implantation method, and all tumor-implanted mice reach morbidity at approximately 12 to 15 days. Dietary drug regimens and body weight. All mice received the standard diet unrestricted, SDUR, prior to initiation of the study. Tumor fragments were implanted on day 0. Upon implantation of the tumor, mice remained on the SDUR until a 15H fast was initiated 72H after implantation. Following the fast, mice were introduced to the ketogenic diet restricted, KDR, or remained on the SDUR, depending on the study group. The KDR has a caloric density of 7.12 cal-G. The percent nutritional breakdown of keto-GEN, Medica Nutrition, Canada, is as follows: 2.1% of cal from carbohydrates, 8.7% of cal from protein, and 89.2% of cal from fat, supplementary table 1. The Medica Nutrition Cancer ketogenic diet that we used was prepared without added vitamins and minerals. Previous studies by Tannenbaum showed that the therapeutic effects of calorie-restricted diets come largely from the restriction of macronutrients and not from restriction of micronutrients. Mice were individually housed beginning at the initiation of the 15H fast. Food intake for the KDR mice was measured to be between 1 to 3 g per day to maintain a 15 to 18% body weight reduction in each mouse. Mice were weighed daily to ensure weight maintenance. Supplementary Fig. Mice receiving the SDUR were given the standard chow diet, lab diet, ad libitum for the duration of the study. For those mice that received DON injections, a fresh stock was prepared and diluted to an appropriate concentration in PBS and was administered intraperitoneally, IP. The DON stock solution in PBS was stored at -20°C for the duration of the study, and mice received 200 L injections of 0.1 to 1.0 mg/kg. Mice in the DON survival study received DON at a dosage that was appropriate for the individual mouse response. Some doses were skipped if the mice appeared lethargic or if body weight loss exceeded 1.5 g from the previous day. Studies were terminated at the time of morbidity for the control SDUR group. Bioluminescence imaging. The Xenogen IVIS system is used to record the bioluminescent signal from the labeled tumors as we previously described. 37. Briefly, for in vivo imaging, mice received an IP injection of D-luciferin, 50 mg/kg, in PBS and isoflurane, 5% in oxygen. Imaging times ranged from 1 to 5 minutes, depending on the time point. For ex vivo imaging, brains were removed and imaged in 0.3 mg D-luciferin in PBS. The IVIS Lumina-cooled CCD camera system was used for light acquisition. Data acquisition and analysis was performed with living image software, Caliper LS. Histology. Brain tumor samples were fixed in 10% neutral buffered formalin, sigma, and embedded in paraffin. The brain tumor samples were sectioned at 5 micrometers, were stained with hematoxylin and eosin, H&E, at the Harvard University Rodent Histopathology Core Facility, Boston, Massachusetts, and were examined by light microscopy using either a Zeiss Axioplan 2 or Nikon SMZ 1500 light microscope. Images were acquired using spot imaging solutions, diagnostic instruments incorporated, cameras and software. All histological sections were evaluated at the Harvard University Rodent Histopathology Core Facility. Immunohistochemistry, Key 67 and IBA1. For immunohistochemistry, the tissue sections from untreated and treated tumor-bearing mice were deparaffinized, rehydrated, and washed. The tissue sections were then heat-treated, 95°C, in antigen unmasking solution, Vector Laboratories, Burlingame, California, for 30 minutes. Tissue sections were blocked in goat serum, 110 in PBS, for 1 hour at room temperature, treated with Key 67 primary antibody, rat monoclonal, DACO, 1:100, overnight at 4°C followed by Alexafluor conjugated antirat secondary antibody at 1:100 dilution. Nuclei were stained using NUC blue, in Vitrogen. Images were captured by the EVOS FL cell imaging system fluorescent microscope. For IBA1, tissue sections were blocked with IBA1 primary antibody, rabbit monoclonal, ABCAM, 1:1000, overnight at 4°C followed by biotinylated antirabbit secondary at 1:2000 dilution. Avidin biotin reaction was complete by incubating the tissues in ABC reagent, Vector Lab, for 30 minutes followed by DAB substrate reaction. Methyl green was used for staining the nucleus. Western blot analysis of IBA1 protein expression. Frozen tumor and normal brain tissues were homogenized in ice-cold lysis buffer containing 20 mmol per liter tris-HCl, pH 7.5, 150 mmol per liter NaCl, 1 mol LNA2 EDTA, 1 mmol per liter EGTA, 1% triton, 2.5 mmol per liter NAPI, 1 mol L-aglycerophosphate, 1 mmol per liter Na3PO4, 1 AgMl lupeptin, and 1 mmol per liter phenylmethylsulfonyl fluoride. Lysates were transferred to 1.7 ml Eppendorf tubes, mixed on a rocker for 1H at 4°C, and then centrifuged at 8100 xg for 20 minutes. Supernatants were collected and protein concentrations were estimated using the Biorad detergent-compatible protein assay. Approximately 100 g of total protein from each tissue sample was denatured with SDS page sample buffer, 63 mmol per liter tris-HCl, pH 6.8, 10% glycerol, 2% SDS, 0.0025% bromphenol blue, and 5% 2-mercaptoethanol, and was resolved by SDS page on 4 to 12% BIS tris gels, in Vitrogen. Proteins were transferred to a polyvinylidene difluoride immobilin TMP membrane, Millipore, overnight at 4°C and blocked in either 5% non-fat powdered milk or 5% bovine serum albumin in TBS with tween 20, pH 7.6, 41 to 3H at room temperature. Membranes were probed with primary antibodies, IBA1, APCAM, UK, overnight at 4°C with gentle shaking. The blots were then incubated with the appropriate secondary antibody, antirabbit, 41H at room temperature, and bands were visualized with enhanced chemiluminescence. Each membrane was stripped and reprobed for actin as an internal loading control, and the ratio of the IBA1 to actin was analyzed by scanning densitometry, fluorochemistry 8900 software. Liquid chromatography mass spectrometry analysis of DON. 2 independent LCMS instruments and procedures were used to measure the amount of DON in the blood and in the VMM3 tumor tissues. This was done to validate the accuracy of the procedures. A triple quad method, Agilent 6460, was used to analyze DON in the first procedure, figure 4a. Mice were injected with 1 mg per kilogram of DON at either 10 or 60 minutes before collection of the brains and blood. The brains were immediately flash frozen. Blood samples were collected before removal of the brain. 3N-HCl plus N-butanol, 250 L, was added to 50 L of plasma, vortexed and then centrifuged at 16,000 xg, for 5 minutes to precipitate proteins. A 200 L aliquot of the supernatant was transferred to a 1.5 ml tube and incubated at 60 degrees Celsius for 30 minutes in a shaking water bath to perform the DON derivatization reaction. For the brain tissue, 5 L of N-butanol containing 3N-HCl was added per milligram tissue. The tissue sample was then homogenized with a glass pestle and was then vortexed and centrifuged, and prepared similarly to the plasma samples. Standard solutions were prepared by serial dilution to generate concentrations from 10 to 100 m. DON was added to untreated mouse plasma or brain tissue to generate an internal standard curve. A high-resolution LCMS Q2F method, Agilent 6550, was used to analyze DON in the second procedure, Fig. 4b. Mice were injected with DON as above. Pre-weighed frozen brain tissue was transferred to a tube containing ceramic beads, Omni tubes. 3M HCl plus butanol was added at a concentration of 5 L mg of tissue. The sample was then homogenized via a bead rupture homogenizer for 45s, let rest for 30s, and homogenized again with the bead rupture for an additional 45s. Subsequently, the homogenate was derivatized by incubating at 60 degrees Celsius for 30 minutes. The homogenate was then centrifuged at 16,000 xg for 10 minutes at 4 degrees Celsius. The supernatant was transferred to a fresh 1.5 ml tube and was centrifuged again to remove excess debris. A 200 L aliquot of the supernatant was dried in a centrifugal evaporator. The samples were resuspended in 50 L DH2O containing 0.2% formic acid. A 5 L aliquot of this solution was injected for DON analysis using a Phenomenex Kinetics 2.6 mF5 column for an 8-minute gradient. Standard curves were also used as described above. Blood glucose and keton measurements. All mice were fasted for 2H before blood collection to stabilize blood glucose levels. Blood glucose and keton levels were measured using the KetoMojo monitoring system, KetoMojo, Napa, California. Whole blood from the tail was placed onto the glucose or keton strip. The KetoMojo meter was used to determine the mole levels of glucose and hydroxybutyrate in the blood. The GKI was determined as we previously described. 36. Tumor necrosis factor determination. VMM3 brain tumor tissue and normal brain tissue from the VMDK mice were homogenized and processed following the Quantikine mouse TNF immunoassay protocol, R&D systems. TNF levels were measured in triplicate and adjusted with the protein level of each sample. The limit of detection is less than 10 pg/ml tumor necrosis factor, TNF. Statistics and reproducibility, body weight, food intake, tumor growth, and plasma metabolite levels were analyzed using the one-way analysis of variance, ANOVA, followed by Tukey's post-hoc test or by a student's t-test to perform a two-sided pairwise comparison among the groups, SPSS 14.0. In each figure, error bars are mean plus or minus SEM and N is the number of individual mice analyzed. The survival studies were plotted on a Kaplan-Meier curve using GraphPad Prism software, and significance was determined using the logrank test. Reporting summary. Further information on research design is available in the Nature Research Reporting Summary linked to this article.